right. Well, based on the popcorn counter, our rate of new people joining has slowed. I apologize. I have a little bit of background noise. I am part of the CFAR team at UAB, and we were doing a site visit in southeastern Alabama this morning, and I'm on my way back from Birmingham, and conveniently in a Starbucks to introduce our Grand Rounds and MHIGH seminar speaker. My name is Lynn Matthews. I'm an associate professor in the Division of Infectious Diseases. I'm also an associate director with the Mary Hersink Institute for Global Health, and we are co-sponsoring this talk today. I'm thrilled to introduce Michael Vinokur. Michael Vinokur is an associate professor in the Department of Medicine and in the Division of Infectious Diseases. He's part of our Global Infectious Disease Research section. Michael did his bachelor's degree at University of Illinois, Urbana-Champaign. He did his MD at Loyola and his residency also in Chicago at Rush. And then I don't actually know the story to this. Someday I got to hear it. Ended up doing ID fellowship at UNC Chapel Hill. And since then has been doing a lot of work that we're going to hear about today in Zambia. And he joined the faculty at UAB and does a lot of work in global infectious disease work. He does translational stuff. He does HIV care, cascade work, and behavioral intervention studies. He does like quite a gamut of work, and recently has been getting more into hepatitis. And today Michael is going to talk to us about hepatitis B cure, and we're excited to hear from you. So take it from here.
Great, thanks so much. Yeah, so I do a lot of eclectic things for an infectious disease doctor, but I think that's kind of a theme in ID that we kind of go where the problems are, even if they don't look like classical diagnosis and treatment of infectious disease. So that's kind of been the story of my career. So today I will cover hepatitis B cure, the state of the science and future direction. So I feel like although we have, several faculty members in the division who have quite extensive hepatitis B research, we don't see a lot of it clinically. A lot of that goes to hepatology, and I don't think it's really a hot topic in the division. And so I think there has been a lot happening in this field, and I just wanted to share that with the other faculty for their awareness, just to keep a little bit abreast of that, because we're all those of us in Hep B are really hoping it becomes a story like the one that happened in hepatitis C over the last decade. And so I'm glad to just give you an update, and then I'll weave in a little story of my own personal project in global health. Okay, so as many of you may or may not know, so as Lynn said, I'm a faculty member in the division, but I work extensively in sub-Saharan Africa, and I do clinical work at UAB Hospital several times, several rotations per year, but the majority of the time I'm off campus. And so I haven't had as many interactions with everyone on this call as I would have liked, but it's always really fun to come back. But please, just because I'm out of sight doesn't mean I'm out of mind. I'm always really happy to interact with everyone in the division. So Lynn mentioned that we have a sort of a loosely formed section of global ID research within the division. So I just wanted to tell you more about that for our own faculty awareness. And I think we also have some applicants to our fellowship program. And so basically, there are about 7 or 8 faculty who are really doing almost full-time research and with a global or a global local focus. And one thing that kind of unites them is that most of them, including myself, have lived outside the country or were born outside the country and have kind of unique perspectives on working with disadvantaged populations in low-income countries. And so as you can see, we mostly focus on sub-Saharan Africa. We have active research projects across a range of topics. You can see there in about 5 or 6 different countries in Central, East, and Southern Africa. And then the other thing I think is important to recognize is that some people are now based in the US after having a long stint in Africa and have started, growing portfolio of projects in public health and research in the local communities in Alabama. And as you can imagine, there are a lot of parallels. And so I feel that this group of faculty, we have unique experiences in working with, non-traditional partnerships and doing research and with hard to reach populations and being creative about different things that sometimes make administrators at UAB upset. So please, for anyone in the faculty who hears this, and if you ever come across any really unusual research operations, public health operations issue, probably someone in this group has had to deal with it, and we're happy to share our experience. And then also a plug for a new scientific working group. So our CFAR was recently refunded for 5 years under Professor Renee Heffron. And so I will, under the new cycle, I'll be leading a group that's focused on global health reciprocal innovations, along with Janet Turan and also Reina Patel within our division. And this group is hoping to forge more collaborations between the local communities in the southeastern United States and in low and middle income countries, and try to translate experiences back and forth under the HIV umbrella. So now I'll move into the main topic, which is hepatitis B. And as a practitioner, a clinician in hepatitis B, I do a lot of training. I do a lot of patient care in Africa on hepatitis B. And one thing that's kind of universal is that in our medical training, we don't get enough information about it. We don't get enough training. It's not a prominent topic. So I felt like it would be a good idea to give some slides of refresher just to bring everyone up to speed on the problem. Again, because it's also not something that we see every day in Alabama, where most of you practice, or in the US. In brief, hepatitis B virus, it's an enveloped virus. It has a lipid envelope on the outside that's represented in this darker blue. And on the surface of that envelope is called the surface antigen protein, which has a medium, small, and large form. And the capsid has the core protein. So these surface and core proteins are important in hepatitis B. So when the virus enters the body through different means, percutaneous, sexual, mother-to-child transmission, it travels in the bloodstream until it meets the NCTP receptor, which is a unique receptor on the hepatocyte or the liver cell. And once the DNA enters into the cell's nucleus, two forms arise. Let me just move my notes here. So the two forms are called cccDNA, or covalently closed circular DNA. It's a mini chromosome which is highly stable for the rest of your life, basically. We have never found a way to get rid of this. And that cccDNA has the genetic material to complete the life cycle. So transcription to RNAs translated to proteins, and that makes mature virions, you can see on the bottom right. So that's the pathway to replicate the virus. In addition, HBV also integrates into the host genetic material. I think a little bit similar to HIV integration that we think about. But what's different is that those integrants, they only produce the RNAs that lead to the production of surface antigen. And so in addition to having the transcription of the reservoir of hepatitis, making new virions, you also get production of a lot of just free surface antigen protein. And that actually is 10 to 100 times more concentration than the actual viruses. And so, as you can imagine, when we screen someone to see whether they have active hepatitis B and need clinical care, we screen for that surface antigen test. So if we find surface antigen in the blood, then we know that this virus's life cycle is active, and it is transcribing from either integrated or cccDNA. And then the other markers of HBV that are commonly used, just as a refresher, are the antibodies to surface antigen, which indicate a prior infection that resolved or vaccination. Antibodies to the core is a current or past infection. And we use the HBV DNA, that's our viral load test, as evidence that there is transcription of the cccDNA pathway with mature virion production. So after the infection, oh, sorry, let me go back to my notes here. So one reason for optimism in hepatitis B cure is that millions of people have actually naturally resolved their infection. So this is, different from what we see in HIV, where we don't have examples of natural cure. And people who resolve their hepatitis B infection end up having negative surface antigen and positive surface antibodies and core antibodies, as you can see above. That's their profile. And this natural resolution of infection is very common in adults without any immune deficiency. So approximately 95% of adults, if you don't have prior exposure to hepatitis, you may have acute infection, but it will resolve within 6 months. So only about 5 to 10% will progress to chronic infection. On the other hand, children, especially infants, young children, and people with immunocompromise, including with HIV, they, if exposed to hepatitis B and infected, they have a much higher rate of progressing on to the chronic infection. And because this virus is very old and adapted to infect humans, there's a huge pool of infections globally, reaching 240 million individuals. And you can see that globally on this map here, the majority of cases of chronic hepatitis B occur in Southeast Asia and Western Pacific. However, there is a pool of 60 to 80 million in the Africa region where several UAB faculty members are working on this. So in addition to myself, there's also Jody Dion, who has research trying to prevent mother-to-child transmission of hepatitis B, which is critical because, as I just mentioned, childhood transmission often leads to the chronic state. And then you can see on the right the contrast between Zambia and the US. So in the US, the population prevalence of chronic hepatitis B is estimated at 0.3 to 0.6%. So that yields about 1 to 2 million individuals with chronic hepatitis B. And in Zambia, where I primarily work, it's about tenfold more prevalent. And so we have a population of 20 million, but around 600,000 people with chronic infection. And in some populations in Zambia, such as men or people with HIV, it can reach around 10 to 12% prevalence. Like many other viral infections, the antiviral immunity is central to the natural history. And so one thing I want to point out, and just refreshing your understanding of hepatitis B, is that it's not the virus that kills or damages the liver. The virus is very well adapted to hepatocytes and replicates without triggering any kind of apoptotic cascade. So instead, the damage, the liver inflammation, and the injury caused by the virus is actually due to the immune response in response to the virus. And so the immune response to HBV is, a lot of people have worked on this, but HBV is relatively neglected. And for a recent proposal, I estimated that HIV receives about 50 times the funding as HBV. And so there are only a handful of really experienced HBV immunologists when compared to HIV, where we have, hundreds of top people working on this field. And so there's still a lot to be learned. But essentially what we do believe is that cellular immunity or adaptive immunity is central. So there's after the infection, the virus and the surface antigen is released, and there are then targeted immune responses to those proteins, and usually comprising of CD4, CD8 responses. And then in addition, obviously through B cells, you have various antibodies that are produced that we use clinically. And then with vaccination, we're vaccinating with the surface antigen, and that generates the surface antibodies. There's also some emerging literature around innate immunity, but that's thought to be more on the local level within the liver, and relatively few studies have studied that inside the liver. It's one area I'm trying to shed light on. Okay. The other thing that makes hepatitis B complicated field, and that leads to problems in getting funding, raising advocacy, a lot of issues, is that it has kind of a complex natural history. So the chronic infection is not static. So things change over time. Levels of surface antigen in the blood, viral loads, and the degree of liver disease change over time, not always getting worse, but sometimes even just getting better over time, getting worse. And there's this, so if you manage hepatitis B, you would know that longitudinal follow-up is really essential, because sometimes a person looks like they're having a very mild infection with low viral load, a normal ALT, and then 6 to 12 months later, now they have a high ALT and a higher viral load. There's quite a lot of dynamism between the immune system and the virus. But overall, there tends to be a natural decline in the surface antigen in blood. And so if you follow people long enough, there's also a rate of surface antigen loss. So even though these are people with chronic infection, they still do resolve at a rate of about 0.5 to 1% per year. And because it's such a slow rate, it's made it really hard to study the mechanisms, like what is actually driving the natural resolution of the infection. And so we really lack the right studies, because you need years of follow-ups, you need the right samples, the right time points, the right clinical characterization. And because the field is generally underfunded, most of the hepatitis B research, including published in the top journals, is cross-sectional. So longitudinal research is still relatively uncommon in this area. And why does the surface antigen decline over time? That's thought to be an immunological process, but again, it's still a question mark as to why that's happening. So what happens when you treat? So many of us have experience using the active treatments against hepatitis B. So that would be tenofovir, lamivudine, and or emtricitabine. And so I know some of us use it in the context of HIV co-infection. Others may have experience with treating HBV alone, HBV mono-infection. So the nucleotide analogs, similar to what they do in HIV, is they block the reverse transcription of the virus. So they block where that X is. And they lead to, as you can see on the bottom right, a reduction in the production of new virions, and the HBV DNA viral load drops to an undetectable level. And that makes the person non-infectious. So from a public health standpoint, people ask me, about what are the benefits of treating or treating earlier. And I say, well, one of them is that by treating someone, you also prevent them from spreading to another person. You can think of scenarios such as people who inject drugs, or with high-risk sex, or pregnant women, where there's huge public health benefits of earlier treatment. But it's important to remember that other pathway, starting from the integrated DNA, because that pathway continues to produce large quantities of surface antigen. So clinically, after initiating treatment, the viral load may reduce, but the surface antigen remains positive. And so especially in clinicians who don't have a lot of experience with hepatitis B, they think that after 1 or 2 or 3 years of treatment, that surface antigen, why is it still positive? What's going on? Is he not adhering to treatment, or is she not taking it? And actually, that's kind of what we expect, because the surface antigen has 2 ways of being produced, through the mature virions and the other pathway. And so the nucleotide analog therapy doesn't actually reduce the surface antigen. The other well-known therapy for hepatitis B is interferon alpha. And so the FDA has approved interferon alpha for chronic hepatitis B, and it's usually given as weekly subcutaneous doses for 48 weeks of therapy. And you do have a response. So there's 15 to 20% will become virally suppressed, and you will actually have 3 to 6% loss of the surface antigen. Remember, the natural rate is about 0.5 to 1%. And these effects are often sustained after you discontinue the interferon. However, just like in the story, a little bit like hepatitis C was, it's really never been taken to scale because of side effects, because of the requirement of weekly injections. And so I think that relatively few patients around the world are actually receiving interferon alpha therapies. So why do we need hepatitis B cure therapies? Why do we need this? So there are a couple of different reasons. Number one is this is a very prevalent and harmful virus. So there are 240 million people who are infected with chronic infection. And this yields about 1 million deaths per year. And to put that in perspective, there have some modeling studies think that now hepatitis B and C have surpassed HIV, have surpassed TB in terms of mortality rates per year. That is quite significant. It puts viral hepatitis into the top 7 or 8 causes of death worldwide, up with cardiovascular disease and TB. So it's quite a prevalent and harmful viral infection. And the other reason that we need cure therapies is that our current therapies are not completely achieving what we want. So currently, as I mentioned, the tenofovir-based therapy will suppress your viral load, which has a lot of benefits, but it doesn't stop the surface antigen production. And so you would require to be on lifelong therapy in most cases. And because the viral life cycle is still ongoing inside of the hepatocyte, there's ongoing risk of liver disease, including hepatocellular carcinoma. And so doesn't fully achieve some of the goals that we would want with hepatitis B. And then finally, our other therapy, as I mentioned, interferon just has a limited role right now. And so in 2019, the NIH published a strategic plan for research to cure hepatitis B. They have 3 general priorities. One is to understand the biology better. And so a lot of what we know about hepatitis B comes from preclinical studies in animal models and cell culture. And so there's still need to sort of understand this in the human context. And when it comes to HIV, there really aren't good models for HIV-HBV co-infection yet. So there's still a lot of basic science gaps. And then strategic priority 2 is to develop tools and resources. So there's need for animal models that can recapitulate the natural history. There's a need for new biomarkers to understand all of the steps in the life cycle. There's need for, preparing clinical trials units, understanding patient preferences when it comes to hepatitis B treatment. There's just a lot to do in that area. And finally, priority 3 is to create strategies to cure and prevent hepatitis B. So this covers the development of new drugs that may block the replication or eliminate the reservoir, as well as other things such as how to set up programs to eliminate the virus. So before I talk about some of the specific therapies that are in the pipeline that I think you should be aware of, I want to define what it means hepatitis B cure, because this has been debated a lot in our field. Complete cure, the first option, is thought to be unrealistic. This is complete eradication of all the nucleic acids of HBV from the body. So elimination of the reservoir. And we're not really sure if this is feasible yet. There have been some studies showing that the reservoir can be degraded, but it's still kind of controversial if this is a real thing that we can achieve with drugs. But the main focus right now is the second one, functional cure. So functional cure is defined as the sustained clearance of the surface antigen, less than 0.05 international units per ml. So like quite a sensitive surface antigen assay, as well as, no detection of the virus in the blood, viral load in the blood. And we're targeting this endpoint because it also mimics what happens in the natural clearance. And there's some evidence which suggests that, sure enough, when you achieve the functional cure, you do have silencing of the reservoir of infection inside the liver. Then at the bottom, there's the term partial cure, because many people who have a very low surface antigen that are just outside of that range, so for example, just over 0.05, but maybe less than 100 international units, over time, they're likely to progress on to developing resolution. And so people would say, let's make the end zone a little bit closer so that we can have success in our trials. So that's a bit controversial, and just to give you I thought I would just give you some perspective, because I think many of you don't measure the surface antigen. These are some surface antigen levels from our cohort in Zambia. And so you can see that this is before people go on to treatment. The surface antigen, the median from this is just a scatter plot. The median was about 13,000. So quite high compared to the levels they need to reach for the functional cure. But you can see on the far left there are a number of patients who are, very close to zero already. And so these are the individuals who, we would expect maybe over follow-up could reach the functional cure. Okay. So preclinical studies have, been really helpful in the last 10 to 15 years to understand HBV biology. So I think there have been a lot of great work in understanding the precise mechanisms and the potential targets for new therapies. And if you had followed the hepatitis C cure story over the last 10 years, it was really fundamental biology, molecular biology, that led to us understanding exactly how the Hep C life cycle worked, and that created targets that could then allow us to develop the DAAs that we have now. And so generally, this is kind of the same approach. And actually, some of the virologists who worked on hepatitis C, won the Nobel Prize for hepatitis C, are really now shifting increasingly into hepatitis B to try to also tackle this virus. Broadly speaking, we think about two different therapies. We have the antivirals and the immune modulators. So I will go over each of these categories and give a couple examples. So here are a couple examples of antivirals. So the first one is called the capsid assembly modulator. And so, as I mentioned, the capsid contains the viral DNA, and so by impacting the capsid, you can slow down the life cycle. Basically, binding to the capsid proteins causes aberrant formation or capsids that don't have the DNA inside of it. And so one potential advantage that capsids have is that you can see here where it says CPAM, they can work on two parts of the life cycle. As the virus is infecting the hepatocyte and as the DNA is moving into the nucleus, and they can also work downstream as the RNAs are then being translated into the proteins that form the capsid. And in the earlier studies of CAMs, there was a lot of HBV DNA suppression seen. However, the levels of surface antigen reduction were not very impressive. And so these are still around and being studied in additional trials. I've now seen that there's some newer CAMs, newer generations, and probably with higher doses that are achieving some surface antigen reduction. And one reason why this drug is still, another reason why it's still being evaluated a lot in cure is that it's an oral agent which has very, very good side effect profile. And so there's also the thought that maybe this could become an alternative to the nukes that we use, because of course there are some people who can't tolerate nukes, and this medicine has a very potent effect on the virus as well. This is the second one, antiviral. They're called ASOs, or antisense oligonucleotides. So ASOs have emerged in the last decades. It's a brand new class of drugs. These are short, synthetic, single strands of nucleic, of DNA molecules. And they alter RNAs. They basically target mRNAs and can, either reduce or restore or modify the expression of a protein. There are already about 13 approved by the FDA for other conditions, like, for example, some neurological conditions. They all have ASOs. And an ASO called bepirovirsen, which is now commonly called Bepi, was designed originally, by GSK and developed by GSK, and it was designed to target some of the hepatitis B mRNAs, including the pregenomic RNA. And in the preclinical studies, it was shown to reduce surface antigen and reduce the viral load, and so it moved into clinical studies. And then in a phase 2b study, which was called the Be Clear study, it went into a randomized trial, which you can see here. In the Be Clear study, the adults with HBsAg of at least 100, so the level of at least 100 at baseline, with or without concurrent nucleotide analog therapy, they were randomized to 4 different strategies, but I want you to focus on the strategy that has the red box around it, because that's the most aggressive strategy in terms of the dosage of Bepi and the duration. And they used as their primary outcome functional cure. And so again, just to reiterate, when functional cure is when your surface antigen reaches a very, very low level, less than 0.05, there's no HBV DNA detected, and you're off therapy for at least 6 months. So this is the way a lot of Hep B trials are designed. And so now let me show you what the results are of that group in red. So you can see that during the Bepi therapy for 24 weeks followed by 24 weeks off therapy, there were some participants who achieved very low levels of surface antigen and even the functional cure. So about 9 to 10% at the primary endpoint actually now had HBV functional cure according to the definition. I think you can see it a little bit better here because I find all those other bars a little bit confusing, but you can see that the number of participants with the very low levels of surface antigen had really increased by N line. And so for us in the hepatitis B field, this was a landmark study, 10% functional cure. And in addition, about 20% of participants had the so-called partial cure. And I think it was a huge landmark because it's really, I think, the first clinical trial showing that over a relatively short period of time, you could achieve functional cure in a meaningful number of participants. But to put that in perspective, 10% can be disappointing. And so I think there's a lot of concern in the field that if we don't get higher rates of functional cure, that there may be a loss of interest by pharmaceutical companies who are investing a lot in this right now. So the last antiviral I wanted to go over is bulevirtide. This is an entry inhibitor. So the NTCP receptor was just discovered in the in 2010, I think. So not that long ago. And then this drug was developed in order to block the receptor, very straightforward approach. And so as this was tested in hepatitis B, there was no reduction, overall in the surface antigen levels. But what they found during those initial trials was that it actually had a potent effect on hepatitis delta. So now I'm actually going to segue into hepatitis delta briefly, which I think is even less discussed in our division because it's just really uncommonly seen. And until now, we haven't even had any treatment. So what is hepatitis delta virus? So hepatitis delta virus is a defective, you can see it on the right, a defective non-enveloped virus that can only replicate its life cycle in the presence of hepatitis B surface antigen. So it needs hepatitis B in order to live. Therefore, it can only infect and replicate in people with chronic hepatitis B. Now, what's the prevalence? Well, there's a lot of lack of, there's a lack of data, so we don't fully know for many geographical areas, but generally it's in the range of 5%. So that means that 5% of the 240 million people living with hepatitis B chronically may have, so that's 10 to 20 million, may have hepatitis delta co-infection. But there's a lot of regional differences. So in Cameroon, for example, in Central Africa, about 12% had hepatitis delta, whereas in Zambia, where I work, we've screened about 1,000 people, I think, at this point, and found like 1 positive. So it's very rare in Zambia. So it seems like there's a lot of need to map the epidemiology. But regardless, if a person has hepatitis B and delta co-infection, this triggers an accelerated course of liver disease. This is really considered to be the most dangerous form of viral hepatitis because of the course of liver disease leading to liver cancer, and also because there's really not been any treatment available up until now. And so bulevirtide, the NTCP entry inhibitor, was studied further and then in randomized trials now published in the New England Journal last year, and its safety was demonstrated. And you can see here that it had a good reduction in the level of viral load, and that also improved the degree of liver disease for those individuals in the trial. So as a result of this and several other studies, the European Medicines Agency did approve this drug for hepatitis delta in Europe, and it's currently under FDA review and approval. And so I think if this gets FDA approval, I'm sure that you will hear more and more about the need to screen, because I think until now, outside of research, I don't think we routinely screen. But if you want to screen and you have patients with chronic hepatitis B in your clinics, you would screen with an antibody test first, hepatitis delta antibody. And I know it's available in some of the private, like Quest and other labs. I don't know if it's necessarily available through UAB. And then if that comes positive, you would need to confirm with the hepatitis delta RNA to confirm. So stay tuned on that. Now I'll move over to immune modulation. This is the second strategy for cure. And so I'll talk about a couple different strategies here. The first one is toll-like receptor agonist. So one of its strategies to retain chronicity in the body is to suppress the toll-like receptor expression levels, which blocking downstream signals to innate immunity. And we've seen that antiviral therapies actually do partially restore this mechanism. And so TLR agonists may activate intracellular signaling. You can see that on the left. So you can see that within the HBV infected hepatocytes, TLR agonists may lead to some antiviral activities. And then also you can see on the right, the TLR agonists may help cells in the region of the hepatocytes to activate immunity through presenting more antigens and activating viral specific T cells. So there's a good rationale to try these drugs in the context of HBV cure. And so actually the ACTG is now conducting a clinical trial, phase 2, of a TLR8 agonist for people with HIV-HBV co-infection. And for this study, the drug is called selgantolimod. It's actually the only HBV cure study for people with HIV that I'm aware of. So that's really exciting because I do a lot of work in HIV. And to participate, the patients need to have both HIV and chronic hepatitis B. They need to have long-term suppression on their current tenofovir-based ART, and then they have to have, a moderate or low degree of liver disease at baseline. And then the other thing I just wanted to flag is, because I just learned about this, is that the ACTG, the AIDS Clinical Trials Group, which is a huge, massive network funded by the NIH, they rebranded, or they renamed themselves Advancing Clinical Therapeutics Globally. And, I hope that that indicates that they're going to expand from just HIV therapeutics to potentially hepatitis B therapeutics as well. They're already doing a little bit, and I hope they'll do more in the future. The next immune strategy is around checkpoint inhibition. So as many of you may know, in several malignancies now, checkpoint inhibitors against PD-1, PD-L1 were effective at reversing T-cell exhaustion and restoring anti-cancer immunity. And so these were then evaluated in HBV as well. And so the immune checkpoints, if you're not aware, are a way that the immune system can put the brakes on an immune response so that it doesn't lead to damage to the body. And chronic hepatitis B is characterized by immune exhaustion, whereby the immune system is less responsive than it should be to the HBV antigens and proteins. So in the above study, you can see that people with HBV who were virally suppressed on their nuke therapy, which is the standard of care, were given nivolumab, nivolumab is an FDA-approved PD-1 inhibitor used for melanoma. And I think there was also a control arm or placebo. And most participants in this small study, this is just N equals 10 in each arm, most did experience surface antigen decline, and then one had a functional cure, which was really interesting. So here you can see on the left, in this PD-1 trial, they actually measure the receptor occupancy for PD-1, trying to confirm or demonstrate that the drug is actually active. And so you can see that on the left. And then on the right, they kind of show the one patient who achieved functional cure. And you can see in that figure on the right, So the y-axis on the left is the level of surface antigen. And then, and so you can see that the surface antigen was around 1,000 to 1,200 when the drug was initially given at baseline, or BL at the x-axis. And then around 3 weeks later, there was a spike in the ALT, the dark black dots, up to more than 1,000. That's a very high level of ALT. And then in conjunction with that spike, the surface antigen levels reduced very rapidly to undetectable levels at around 12 weeks. And so this picture is also kind of what we see in acute hepatitis B, where acute clearing hepatitis B, there's a very productive immune response, which clears the virus very rapidly, but a lot of times leads to a very high ALT. And so this one patient and this one study just really set off a huge amount of excitement in PD-1 inhibitors in the hepatitis B field. And then the third immunotherapeutic approach is therapeutic vaccines. So we know vaccines very well for preventing, used to prevent infections, but there's also therapeutic approaches that involve vaccines. And so just as a background, if you give a person with chronic hepatitis B a hepatitis B vaccine, maybe you didn't know that they had chronic hepatitis B. That vaccine is based on the surface antigen, and it does not elicit any change to their situation. It doesn't cause their viral load to go up or down. It has, really no detectable response. So those vaccines the current vaccines can't be used for hepatitis B cure, but the newer vaccines are using different antigens or additional antigens to make more immunogenicity. And also they're really trying to target T cells and create HBV-specific T cells that can, lead to an immune response. I think there's one big challenge, which is that the vaccines really need to overcome the chronic immune exhaustion state that characterizes hepatitis B. So if you give these new vaccines to a healthy individual without hepatitis B, they have a very robust response. But in someone who's had chronic hepatitis B for 10, 15, 20 years, they seem to be less effective. So this is still a huge area with a lot of work being done by vaccinologists. So therefore, building on all of these successes and challenges, many trials that are starting now or being planned are looking into combination therapy. So this is kind of thought to be the future. So the idea that we need to target the virus in different ways and give these drugs in different sequences in order to find a scenario where we can achieve the functional cure in a large number of people. So for example, you might want to start with something like Bepi or a CAM, which will lower the HBV antigen load. You can see that on the left. And then maybe you would give a PD-1 inhibitor followed by a vaccine. So you kind of mix and match these strategies in order to find one that can effectively allow the immune system to take control of the infection. And so this is the cohort that I've been managing for a couple of years in Zambia. It's called the HepMech cohort, funded by an NIH R01. And I like to think about it as a possible model for this combination therapy approach. In the middle here, you can see that, conceptually, I work a lot with HIV/HBV co-infection. And when someone initiates antiretroviral therapy for the first time, they are receiving, there's kind of a direct and an indirect benefit on or effect on HBV. So there's a direct effect, which is the tenofovir inhibiting the viral life cycle, as we described. But then patients also experience immune reconstitution syndrome with the CD4 restoration and, presumably improvement in a lot of other avenues of immune function. And so these two, we're using both an immune and a antiviral approach when we treat HIV/HBV co-infection. And actually, anecdotally, we do see more episodes of functional cure after treating co-infection than we do in people without HIV. And so this kind of formed the premise for this study. And so in this study, we enroll people with HIV, HBV co-infection, and also HBV mono-infection. And at the point of enrollment, which is before or at the time when they're starting treatment, they undergo a blood test and a liver fine needle aspirate because we want to try to understand what's happening at the level of the liver. And then during, everyone goes on to the standard treatments. And 1 year later, we repeat those research assessments so that we can try to see if we can learn about the immune reconstitution and how that manifests in terms of anti-HBV activity. And then we have a follow-on grant, also working with this cohort that was just funded last year to make a more of a deep dive into the HBV-specific CD4 T cells, because as you would know, the CD4 is heavily affected by HIV. And so it's recently been recognized that it probably plays a really important role in HBV as well. This is a picture of some of my teammates in Zambia, and you can see our little research area where we do hepatitis cohort, and we painted it green correspondingly. All right, I just have maybe 2 more slides. Well, actually, one more slide, actually, and it's to talk about novel biomarkers. So the HBV cure field focuses a lot on new drugs, but in addition, there's also work happening in the biomarker space because the tests that we use currently are a little bit limiting. So right now, we measure the viral load, HBV DNA, and we measure the surface antigen. But once someone is on therapy, and their viral load is undetectable, we really can't tell if we're making any changes to the viral life cycle using those biomarkers. And the surface antigen, as we talked about, doesn't really change very much. So we're kind of looking for different assays which can show the dynamic changes to the life cycle expressed in a blood sample. Because, although I'm doing some liver biopsies for my research study, this obviously can't be scaled to all patients in the clinic. And so you can see on the right, there are various new biomarkers, being used in research type studies. Abbott, Roche, a lot of the big diagnostics companies now have dedicated teams working on these markers, validating them across different populations. And then clinical trials are often measuring them in conjunction with the standard markers to try to see if we can find a marker which might indicate that someone is on the pathway to a cure or might be not responding to the drug, so we can discontinue it, or just to indicate that over time we can monitor patients more intensively than we do right now. Okay, so just in summary, I hope you learned a lot. I hope you learned that nukes, most prominently tenofovir, they suppress the HBV DNA in a person with chronic hepatitis B, but they don't really alter the reservoir of infection, and HBsAg levels remain relatively stable. Lifelong therapy with tenofovir, although we use that in HIV, it's not ideal. We would love to be able to get to the functional cure. And that's where the HBsAg is cleared and you no longer need to have the person on therapy. And so that's really the central focus of the cure field right now. So we've had a lot of improvement in the last decade, and a lot of new scientists now working in this area to try to understand the life cycle and understand the immunology. And in addition, the pharmaceutical companies, many of which who worked in hepatitis C, have shifted over to work in hepatitis B, which has brought a lot of excitement in the field. And there's also a lot of work in the biomarker space. We've seen that with single drugs, either immune modulators or antivirals, that you can achieve a functional cure in like a small fraction of participants. Remember in the New England Journal of Medicine article, only 10% really achieved functional cure, which is exciting for us, but may not be exciting for all people. And so now the push is to move into combination therapies to really try to achieve a higher rate of functional cure in the future in our trials. So thank you very much for all of your attention. I just wanted to acknowledge my, really helpful and close friends of mine at this point after a decade of work, the HepMAC cohort team in Zambia, comprising of several physicians, statisticians, nurses, laboratory technicians, research assistants from the community, some with people with lived experience. So it really takes a village to do global health work, well, any work, but specifically my work takes a village, and I really appreciate the interdisciplinary nature of our team. And then I also couldn't do it without my team, the Global Health Infectious Disease Research Unit Core at UAB, including Jamie and Brittany, and recently Ike joined, and Tawanda as well. And then I have some great collaborators in this work with Hep B immunology at Massachusetts General Hospital, including Ray Chang, Nadia Alatrakchi, and Georg Lauer. And I work closely with Debika Bhattacharya around HBV clinical trials. So thank you very much. It looks like I managed to leave a few minutes for Q&A. Thank you.
Thank you, Mike. That was amazing. And thank you for taking us through the basics all the way to the future. Folks can ask questions. You can type them in the chat. You can raise your hand. You can just go off mute and speak. I will see if I see hands up.
I see Mike Sag.
Go for it.
Turned on his video.
Yeah,
Hi, Mike.
Hey, how are you? Great job. So, I'm a little confused about the bulevirtide and when to use that. It's not approved in the US. Okay, fine. But let's assume it is. And you've got somebody who's co-infected with hepatitis D. So we see the levels come down, but does that correlate to clinical benefit as far as you know? Is it something we should do, is what I'm asking.
Yeah, I mean, I believe that the viral load, driving down the viral load drives down the liver damages. So therefore, from my understanding, it should. I think we expect that it will alter the natural history of disease. And it would be better to drive down viral
We're
load.
not really testing for hepatitis D much in people that have Hep B, and maybe we should in the long run. I have equipoise.
Actually, as I was preparing this, I was looking for like CDC guidelines, or there isn't much yet, but that's why, in Europe right now, where they now have this approved, there's a huge push to screen. So as you can imagine, the drug companies making the drug are encouraging everyone to screen. I mean, in Africa, we received a grant to screen in Africa. We said, well, we don't even have testing for hepatitis B, but you'll give us money to test for Delta. Okay, we'll do it.
Okay, thanks. Well done.
Thank you.
Mike, Stephanie Sabaj asked, what about efforts to implement HPV vaccination? How are things going on the prevention side?
Yeah, I think so. The key group that we're trying to vaccinate comprehensively are newborn babies. So in Africa, most parts, we don't have an extra birth dose vaccine. And so we see, in children who don't have the birth dose vaccine, in some places there's a 1% prevalence. And so we're trying to get that down to 0.1%. That's the elimination target. And so there is a lot of work around providing an extra dose right at the time of birth. But then for adults, it's really remaining very patchy. So there aren't a lot of advocates to vaccinate adults who are naive to hepatitis B. I mean, we do it for health workers on a limited basis, but yeah. I think most of the reason we focus on children is that that's the group that generates the most new cases. So I think there are only about 1 to 2 million new cases per year compared to the 240 million existing cases. So good question. I see another question. After the immune treatment with high LFTs of 1500, does the patient need liver transplant or death? Yeah, so interestingly, like for example, in acute hepatitis B, when you see a patient with an ALT of 2000 or 3000, I've seen, and they're very jaundiced, generally the mortality is extremely low. We don't even have to treat the virus per se. We just do supportive care, and we have resolution most of the time. And even with hepatitis flares that happen, like immunological flares, like that patient, there is kind of natural resolution. It rarely leads to transplant or death. The one time when a flare is very dangerous is after stopping therapy, especially when the person is living with cirrhosis. They can push them over into liver failure. But I actually don't have a lot of experience with transplant because it's not available where I do most of my work. But I have seen in the US a couple acute hepatitis B that had to have a transplant. So it does happen. Thanks for that question.
Mike, I don't see other hands or questions in the chat. While people are thinking about those, maybe I'll ask, since we have fellows today, and this is also co-sponsored by the Global Health Institute, where we're really interested in more opportunities for trainees to get involved in this kind of research. Can you talk about how you engage trainees in your work?
Yeah, sure. So, I mean, we have well, I think first of all, it helps that about half of our group is in Birmingham at any one time. And then those who are overseas are cycling in and out. And so we have really good communication, regardless of our physical location. And over the years, we have mentored several fellows, and we're actually really, I think this is a strategic thing that we want to grow the young investigators in this space, because I think people like Lynn, myself, Jody, we were all new faculty just a few years ago, it feels like, and now we're a little bit more established. And so we want to try to, get the pipeline running again. And so I think for interested fellows, there are a lot of different ways to get involved. You don't necessarily have to be overseas, because a lot of the world is all connected now. And so there are opportunities to write papers, analyze data, get involved in implementing. There's a lot of calls where you learn how global health research is done. And then on top of that, there are opportunities to go into the sites. I mean, we have had fellows over the last, I've been on faculty for about 8 years. I think we certainly have had 2 or 3 fellows that spent a significant amount of time overseas as part of their fellowship. And I think that is very much something that we can work on supporting. But I think if it's important to you, it's good to talk about it early so that those plans can be made. I mean, when I started fellowship, I kind of hoped that the 3rd year I could go overseas. And so I already kind of started planning in the 1st year and trying to figure out which sites we had affiliations and who I could work with. And then by the time I got into the 2nd year, I was starting to think about what would be my project, how would I fund it, et cetera. So with global health, advanced planning is always helpful. But the nice thing is we have now a great group of faculty who are, doing really deep global health work and can figure this stuff out. Sorry, that was a long-winded one.
That's great. Well, we are just at a minute before the end of the hour, so I don't see hands. If I'm missing them, please speak up. I don't see stuff in the chat. Michael, thank you for sharing your work and your thoughts. And thanks everyone for joining today.